Assalamu alaikum and Bismillahir Rahmanir Rahim. So today we have the topic of erythropoiesis, and before starting erythropoiesis, we have recall about hematopoiesis. That hematopoiesis is that process in which proliferation, maturation, and differentiation of all the cells occur, and these all cells are erythrocyte, platelet, and white blood cell. And now today we are going to discuss erythropoiesis. My name is Tanvir Tara and I am lecturer at Sarhad University of Science and Information Technology. This topic is for both 4th and 6th semester. So moving toward erythropoiesis, here you are looking toward this picture, and this picture shows us hematopoiesis. Hematopoiesis divided into thrombopoiesis, leukopoiesis, and erythropoiesis. This is our topic today. And then leukopoiesis is further divided into granulopoiesis and agranulopoiesis. Agranulopoiesis are monocyte and lymphocyte, where granulopoiesis are eosinophil, basophil, and neutrophil. So this is leukopoiesis. Our next topic is leukopoiesis. Today we are going to discuss erythropoiesis. So here on this slide, we have a diagram, and here you know that this is the first pluripotent stem cell which is going to mature, and this maturity is like it goes into myeloid series, and here it is a lymphoid series. So the pluripotent stem cell, which have the capability of self-renewal, it has also the capability of going in further division, and that are two type of division: lymphoid stem cell and myeloid stem cell. So lymphoid stem cell is then further divided into lymphoblastic stages, and like it produce three type of lymphocytes: B lymphocyte, T lymphocyte, and natural killer cells. So this is totally different lineage, and it's called lymphoid lineage. Where on the other hand, on the left side, we have colony forming unit, granulocyte, erythrocyte, monocytic, and megakaryocytic stem cell. And now here, from here onward, it is going to gametid, and it's here that birth forming unit, it goes further and divide into and mature into the red blood cell, where we have platelet here, and we have also neutrophil, eosinophil, and basophil with the monocyte as well. So this is the chart, and we have all the cells production from this pluripotent stem cell. In further detail, in further lecture, we will discuss how these regulation occur, like how pluripotent stem cell goes toward lymphoblastic stage, and how it will goes toward the myeloid stage. There are certain cytokines or interleukins which promote these cells to maturation, and from this slide we learn that the main stem cell, that is pluripotent stem cell, has two types, that are myeloid and that are lymphoid. Two types mean that it's divided into two types, and then we have these cells. So on this slide, let's start erythropoiesis. Erythropoiesis is the process of development, differentiation, and maturation of red blood cell from primitive stem cell. And whenever the pluripotent stem cell is going to mature here, so it will goes toward the maturation, that is maturation of red blood cell, and this is known as erythropoiesis. What happened during this erythropoiesis? During this time, four mitotic division usually occur, and whenever these four mitotic division occur, each subsequent division is accompanied by nuclear and cytoplasmic maturation. They are commonly referred to as maturational division, and each successive division yields a smaller cell in which nucleus become progressively smaller and more compact, and even it become pacnotic until it is ejected or it is extruded from the cell in the final stage, that is orthochromatophilic stage. We are going to discuss it. And usually 6 stages in the development of red blood cells are recognized, that are that is pronormoblast, which is also called erythroblast, then basophilic erythroblast, then polychromatophilic erythroblast, and orthochromatophilic erythroblast, and then goes toward reticulocyte and mature red blood cell. Now we have the idea that how erythropoiesis came into being, and that is the first pluripotent hematopoietic stem cell, then it goes toward common myeloid progenitor cells, then colony forming unit, that is of erythrocyte series, burst forming unit, that is also for erythrocyte series, because from here onward we have committed stem cell, and that commitment is with erythrocyte, and now it will further goes into proerythroblast, then basophilic erythroblast, polychromatophilic erythroblast, orthochromatophilic erythroblast, and finally reticulocyte and erythrocyte. From here up to here, we have the erythropoiesis that take place inside the bone marrow, because these cells have a large size. Here in this diagram you see that the size of the cell is very large, that is unable to come in the stem cell and peripheral circulation, and these are the stem cell, because whenever we are seeing stem cell in the peripheral circulation, then we have some defect or some disease. So normally we can't see these stem cell or these immature cell in the peripheral circulation, but from here onward, that is reticulocyte or erythrocyte, they are then migrated toward the peripheral circulation, and these erythrocyte are mature cells. The erythrocyte, the name itself called that it is a mature cell, because we can't say that this is a mature cell, but whenever we are seeing erythrocyte, so the erythrocyte word itself, it shows that it's a mature cell. Now we have different nomenclature as well, like CAPE and ASCP. So CAPE is the College of American Pathologists, and where ACP is American Society of Clinical Pathologists. They are using different nomenclature for their staging. Like in CAPE, we have pronormoblast, then basophilic normoblast, polychromatophilic normoblast, orthochromatophilic normoblast, and reticulocyte, and then finally erythrocyte, where ACP is using different nomenclature. They are called rubriblast, rubribrow red cells, so that's why it's called rubriblast. Then proribrocyte, then rubricite, metarubricite, and then these two, reticulocyte and erythrocyte, here and in CAPE as well, they have the same nomenclature. So the two different nomenclature is used in erythropoiesis, because in erythropoiesis we are not only seeing the nomenclature of CAPE and ACP, but we are also seeing some different nomenclature as well. Like common nomenclature is proerythroblast, then basophilic erythroblast, polychromatophilic erythroblast, and orthochromatophilic erythroblast, where some mixed nomenclature is that we are also seeing in books as well. So proerythroblast, early normoblast as well, basophilic erythroblast is also known as early normoblast, then intermediate normoblast, and then late normoblast. So in some books we are following this stage. Some books are like mixed nomenclature, and as we have discussed CAPE and ACP, so some are using this nomenclature. But one thing to remember, that when you are using common nomenclature, like you are starting from proerythroblast and then basophilic erythroblast, then you will go forward to polychromatophilic erythroblast. Here you cannot use intermediate normoblast, or then you are using polychromatophilic normoblast. So here one thing to remember, that whenever you are using the normoblast, use the same as it is, like common nomenclature, it will come toward here, In mixed nomenclature, it will like proerythroblast, then early, then intermediate, and then late. In K, basophilic, polychromatophilic, and orthochromatophilic. But here the word is normoblast. If you are seeing here, the word is erythroblast, and here they are the same, normoblast. But here one thing to remember, that here it is early, then intermediate, and then late. And ACP, it's this nomenclature is not so common, but we know that rubri is used for red blood cells, and they are using this nomenclature like because we are using such nomenclature in leukopoiesis, that we have myeloblast, then promyelocyte, then metamyelocyte, and myelocyte. So here they are using such type of nomenclature: rubliblas, then prorubicide, then rubricide, metarubricide, and then we have reticulocyte, and finally we have erythrocyte. So reticulocyte and erythrocyte are the same nomenclature, but here these nomenclature are different from each other. So let's start the stages of erythropoiesis, and here you are watching this, or you are looking toward this diagram. So this is proerythroblast, and then it goes toward, it further divided into basophilic erythroblast. This is day first, this is day second, and on day 3 polychromatophilic erythroblast. Here you are seeing that the cell size is larger, then it's is going toward its maturation, and its size is now somewhat reduced than the first stage. And here you are seeing basophilia, so you are seeing cytoplasm. Here we have nucleus, and then further in this stage, that is polychromatophilic erythroblast, the size become more reduced, and now here the color of the cell is also changed. Here the nucleus become very pycnotic, and here you are seeing the nucleus, it's open chromatin, it's large, NC ratio is high, but here the nucleus is pycnotic, and now the function of nucleus is no more required here. So on this stage, nucleus is become extruded, and it's ejaculate from here, and then the final stage, that are reticulocyte and erythrocyte. So from day to fifth, from day 5th to 7th, reticulocyte enter the circulation, and then it become mature. So erythropoiesis have 6 stages. The first stage is proerythroblast, which is largest cell. It has the size of 15 to 20 micron. It has basophilic cytoplasm, and it has a big nucleus with the open chromatin. And open chromatin mean that it has nucleoli, and it has the capability, or it has the capacity, or it has the ability to mitotic division. And then the second stage, that is erythropoiesis, that is basophilic erythroblast, and here the size become reduced, that is 14 to 17 micron. Mitotic division is still here, and it has still basophilic cytoplasm, because basophilia shows that it is an immature cell, and it has mitotic division. Nucleoli are present here, but in some cases nucleoli are going to disappear, and few nucleoli are still present here. Then on the third day, we have third stage as well, and that is known as polychromatophilic cell, and it's also known as polychromasia. Polychromasia or polychromatophilic cell is that cell in which hemoglobin appear for the first time, because hemoglobin synthesis is still start from here. We have heme synthesis and we have globin synthesis, but it appear on this stage, and that's why it's known as polychromatophilic cell. Because of its color, it's known as polychromasia, because it has some bluish color, and also we know that the color of hemoglobin is red, so it has double color, and that's why it's known as polychromasia. Nucleus become condensed, and here the function of nucleus is now getting reduced. So the next stage is that is normoblast, or it's also called orthochromatophilic erythroblast. So the nucleus become very pyknotic and no function is required more here. Its size become reduced, that is 7 to 10 micron, and extrusion of nucleus occurs here, and on this stage hemoglobin is maximum. Then reticulocyte, and reticulocyte is a little bit larger than the mature erythrocyte. We have some reticulum of basophilic material in the cytoplasm. In the end we will discuss it in more detail, and then we have the final stage, that is that is erythrocyte, and its size is up to 7.2 micron. It's a mature cell and now it's ready for its function. That is, it has the capability of putting oxygen in itself, and then it's move forward to all the body. So these are the stages of erythropoiesis. So we have 6 stages of erythropoiesis: proerythroblast, then basophilic erythroblast, then polychromatophilic erythroblast, normoblast, and reticulocyte, and then finally we have mature red cell. So now we will discuss one by one. This is proerythroblast. This cell is proerythroblast, and you are looking here that it is a large cell. Its NC ratio is high. It has basophilia, and mitotic division is present here. No hemoglobin is present here, but the process of hemoglobin synthesis is start here, and its typical picture is this one. It has air-like projection and a perinuclear halo. That is a clear area around the nucleus, and it's known as perinuclear halo or perinuclear cytoplasmic vacuolation. Its size is 15 to 20 micron, nucleus with multiple nucleoli, basophilia is still present, mitotic division is still present. A basophilic erythroblast or early normoblast. So the size become reduced. You are looking at the previous slide. The size was much more, that is 15 to 20 micron, and now the size is 14 to 17 micron. Nucleoli reduced in number, and basophilia is still present here because the mitosis is active here. And then we have polychromatophilic normoblast or intermediate normoblast or polychromatophilic erythroblast. So here you are looking that this color of the cytoplasm is somewhat different. It's not basophilia, but it's a polychromatophilic color. It means that it is made of two color, that is basophilia and red color. So it give a different dye, and nucleus is going to condense. Nuclear material is now getting reduced, and chromatin clumps are present here. Hemoglobin start first time appearing here, and mitosis now become reduced. Normally no mitotic division is present here, but in certain cases it is still have the capability of the mitotic, but in reduced form. Now, orthochromatophilic normoblast. So, orthochromatophilic normoblast is that cell in which the nucleus function is no more required, and now you are looking in this slide that it has very polychromatophilic color, and now its color is going to become very pinkish. It's mean that hemoglobin is present here, and nucleus is now extruded. So nucleus is extruded here. Its size is 7 to 10 micron. Acidophilic erythroblast, which is the last precursor with a nucleus. Remember that this cell is the last cell which has nucleus. Normally we don't have nucleus in the erythrocyte. So nucleus is compact and situated near the membrane. Pyknotic nucleus is extruded, and cytoplasm is like mature red blood cell reflecting a high hemoglobin content. And whenever nucleus is going to extrude here, so there is not mitotic division is there is no mitotic division, so mitotic division is absent here. and this one is reticulocyte. So reticular nuclear fragments are here. This one is nuclear material, and this is reticulocyte on supravital stand. Because vital stand is required for this stage, because a reticulocyte stand measure aggregate of residual ribosomes and mitochondria that form clumped granular material that is known as a reticulum. So reticulocyte appear is polychromatophilic cell seen on right, or Giemsa stain on blood film. And whenever you are seeing it on the normal Romanowski stain, so it look like this. This is a large cell, slightly larger than the red blood cell. So reticulocyte it has reticular nuclear fragments. Nucleus is extruded from here, that in the previous stage, the chromatophilic stage, and slightly larger than the red blood cell. Its normal count is 0.5 to 2% of all erythrocytes, and then we have mature erythrocyte. This is mature erythrocyte. They are reddish in color, circular, and biconcave cells. They have very flexible shape. Its size is about 7 to 8 micron, but normally we are seeing 7.5 or 7.2 micron. No visible internal structure. There's nothing here but only hemoglobin, and that's why it has high hemoglobin content. A large number of molecules of hemoglobin are present over here. Bright at the center due to biconcave shape. Here you are seeing that this is center, and it has brightness, and all the hemoglobin are here. So duration of erythropoiesis, and while looking at the duration of erythropoiesis, we have hematopoietic stem cell, and from hematopoietic stem cell to erythrocyte, it will take about 21 days, and then it differentiation phase, that is from pronormoblast to reticulocyte, it will take about 4 to 5 days, and then its maturation stage, that is maturation from reticulocyte to erythrocyte stage, so it will take only 2 days. So this is the duration of erythropoiesis, and it's all the process take place about 21 days. Last stage, that is from reticulocyte to erythrocyte, it will take only 2 days. Now, while learning erythropoiesis, we come to know that erythropoiesis is the process of differentiation, maturation, and proliferation of these cells. And these cells are proliferated from the first cell, that is known as hematopoietic stem cell, and from hematopoietic stem cell to the last stage, that is erythrocyte, it will take about 21 days. During these stages, we know that first we see the size of the cell was very large, that is 15 to 20 micron, and this size become reduced, and it we have a mature erythrocyte of 7.5 micron. So change during erythropoiesis, the first is decrease in size, then loss of mitotic activity, basophilia and nucleus are getting low, and whenever the nucleus is extruded, mitotic division completely finished from these cells. So mitotic division is slowly and gradually reduces, because no function is required then, and hemoglobinization. Like in the first two stages, we are not seeing any polychromasia, but in the third stage, we are seeing hemoglobinization, and HB appear for the first time in the polychromatophilic cell or intermediate normoblast. Change of cell shape as well. Like the first stage, we have erythroblast that has air-like projection, that has a perinuclear halo, the nucleus is large, it has nucleoli, it has open chromatin, and then slowly and gradually the size become reduces, and at the end we have a very smart, delicate, flexible, biconcave, red, circular cell that is known as erythrocyte. Disappearance of nucleus, mitochondria, and RNA. At the end, in reticulocyte stage, we have some endoplasmic reticulum, remnants of RNA, but then we have a very clear cell, that is erythrocyte. So nucleus and mitochondria and RNA are disappearing with the passage of time during these 21 days, and then change of staining. If you look, the first cell, that is completely basophilic, and that's why it's known as basophilic erythroblast, and then it goes to eosinophilic or purplish or pinkish color, like reddish color. So this has the changes during erythropoiesis, and inshallah I hope that you have learned erythropoiesis. It has some more important point you have to remember for the examination as well. So thank you very much, and I hope that you have learned it and you will watch the full video. Inshallah, we will meet on the Google Meet on the day as scheduled, and we will discuss in further detail, inshallah. So thank you very much, and keep watching the videos.